DNA carries the genetic instructions. Without DNA, you are nothing. Genes, those are coded DNA instructions. Let me tell you about their function. Proteins in the cell, they control that production. Now let's talk about Griffith's experiments. He suggested that bacteria is capable of transferring genetic information through a process called transformation. Watson and Crick, yeah, they discovered the double helix. Watson and Crick, yeah, they the realist. DNA's got two strands that wrap around each other. Your genetic info, you get that from your father and your mother. DNA is made up of deoxyribose, a phosphate group, and a nitrogenous base. A is for adenine, thymine's got the T, C is for cytosine, and guanine's got the G. These are the bases that make up the DNA inside you and me. Hydrogen bonds hold these pairs together. A pairs with T, C pairs with G, there's no bond better. A and G with two rings are purines, C and T with one ring are pyrimides. DNA is a long molecule that's made up of nucleotides. The molecule is 1.6 millimeters, so it's packed in the cell real tight. Let's take a little spin. DNA and protein tightly wrapped together, that makes something called chromatin. The protein that the DNA wraps around are histones, and together they form nucleosomes. Before a cell divides, it duplicates its DNA. You could say that it replicates. The DNA molecule splits into two strands, and it makes two new complementary ones. They follow the base pairing rules. They aren't having any fun. DNA polymerases make up the DNA molecules by assembling the nucleotides, bringing them together like Bonnie and Clyde. Now let's change the subject and talk about RNA.
Thirsty to talk about the messenger RNAs and message to the cell are not ready, but to talk about ribosome RNAs combines the proteins and make ribosomes. But wait, there's more. That RNA is in charge of making proteins that you can find in my genes. The RNA polymerase separates the DNA strands into concentric rubber bands. Let's talk about introns, exons, and codons. Introns don't code the proteins, but they interrupt the sequence of the gene. Exons code the proteins and peptide sequence. But that's not where it ends. Codons are nucleotides, and they code my genetic trait. They specify a single amino acid that's fantastic, like a proline, glutamine, serine, and lysine. Instead of thymine, RNA has uracil, and you can find it in my cells. Mutations are changes in the genetic material. I hope you enjoyed this video.